right, in this lecture segment, we're going to look at how water diffuses through a membrane. The cytoplasm, the inside of the cell, the cytosol really, is the aqueous solution of the cell. And so water is the solvent, and anything that's dissolved in the water are the solutes, and the combination of both makes the solution. So solvent plus solutes equals solution. Now, if you have a cell, I'm going to draw a cell here just because I have a little space. Let's say that that represents the cell membrane. Then out here, this is the extracellular side of the membrane, extracellular and intracellular. If you have different amounts of solutes on one side of the membrane compared to the other, then you also have different amounts of solvent. And so just like the solutes, the particles will move according to their gradient, higher concentration towards the lower concentration area. Water will also move along its gradient. So we are going to look at some examples where I give you some practice, where I give you some concentrations of solutes and solvents inside of a cell and also outside of the cell, and then we'll talk about which direction things move. But before we do that, let's look at this U-tube. This is imagine like a glass tube, and it is shaped like a U, and so they call this a U-tube. And let's say that you can imagine this is glass, and then they have placed, blocking the tube, right here in the middle, they have placed a circle of membrane. This is not a real cell membrane. It's like a circle of plastic, a special type of plastic that has microscopic holes in it. And it mimics the action of a selectively permeable membrane. And the size of the holes that are in that plastic allow some things to move through because they are smaller than those holes, and prevents larger molecules from moving through. So imagine that you have on this side of the membrane, you have pour into this side a solution that has water as the solvent. So water is being shown just like you're used to it being shown like this. These are the water molecules. And these bigger things, those are some kind of solute. In this example, it's urea, but it really doesn't matter. It's a larger molecule that's acting. That is the solute. So the solute is bigger, and the solute in this example cannot get through the holes in this membrane here. And so what will happen is on the other side of the membrane, they have poured in a solution that has less of the solute. So you're supposed to see that concentration difference in the picture. Somehow the artist has to show you what that starting setup is. So they're trying to indicate that on the left side, there's more solute. On the right side, there's less solute. So less solute here, more solute on this side over here. Now, if the urea, the solute, could move through the membrane, it would want to move from its area of higher concentration to its area of lower. So the urea would want to go this way. It would want to go through to the other side until there was equal amounts of urea on both sides of that membrane. But the problem is the urea can't move because the holes in the membrane aren't big enough. At the same time, the water molecules, if there's less solute on this side, that has to mean there's more water. So there's more solvent in terms of concentration wherever there is less of the solute. And there is less of the solvent comparatively on the side where there is more solute. And so just like the urea will want to go from its area of higher concentration to its area of lower concentration, the water molecules will also want to go from their area of higher concentration towards their area of lower concentration, which you'll notice is exactly opposite. So the way that the solute wants to go by diffusion will be the opposite of the way that the solvent wants to go. And the problem here is that the solute is not allowed to move, but the solvent is allowed to move. And so the solvent will move, which is very interesting. The water will move. I'm going to erase all this. If you set up a U-tube like this, and you have a membrane that only allows water to move, but not the solute, then what will happen is the water will move. It doesn't know that the other component can't move, and the water will move from, in this diagram, the right side to the left side. And this arrow here is showing what will happen to the level of liquid over time on this side of the tube. It will actually go up. It was the weirdest. You can run this experiment, and it is odd how it will go against gravity. It will rise up on one side because the water is moving towards that side until both solutions have an equal concentration of water and urea. So the movement of water gets its own name. The diffusion of water, more specifically, is called osmosis. So it follows the same rule that any diffusion follows. It's the movement of the diffusion of water across a membrane, and it always goes from its area of higher concentration towards its area, and by that I mean water's area of lower concentration. And the concentration of water is always inversely related to the concentration of solute. All right, so let me put in, I'm going to insert a whiteboard really quick. Let's see if I can do this. I'm just going to insert a slide. Okay. All right. All right, so let me draw you some examples here. So what I usually do is I pretend like we have a cell in a beaker. And the cell represents any cell, so that's the membrane. And the beaker solution, this represents whatever the solution is outside of the cell. Of course, in the body, there would be an outside solution and an inside solution, but I just use this setup, and this is a pretty common setup for teaching the fundamentals. So this is my cell. It's in a beaker solution. The cell solution is the cytosol. So the cell solution, the cytosol is inside the cell. The beaker solution is all of this stuff outside the cell. All this, okay? So when I say beaker solution, I'm talking about what's outside of the cell. And when I talk about the cell solution or the cytosol, that's what's inside the cell, okay? And so I'll set up a scenario, and you're going to tell me what is going to happen, okay? Now, this is always going to be diffusion and osmosis, all right? So movement of the water from its area of higher concentration to its area of lower, and movement of, I'm trying to get rid of that, sorry. Movement of the solute from its area of higher concentration to its area of lower. Let's see if I can get, okay, I'm going to get a different color on my pen. Maybe I am, maybe I'm not. Okay, let's say inside this cell is 10% starch. 10% starch. I'm only focusing on one kind of solute. And let's say outside of the cell is 50% starch. So you remember starch. It's a polysaccharide. And so there is more concentration of starch outside the cell and less concentration of starch inside the cell. But starch is the solute. Starch is the solute. So if the starch could diffuse across the membrane, it would go from its area of higher concentration, 50% is higher than 10%. So it will go from the outside of the cell into the cell until both sides of the membrane have an equal concentration of starch. However, let's say that the starch cannot cross the membrane. It's too big. So typically I'll set up a scenario where I'll tell you the concentration of the solute, but then I'll tell you that the solute is too big to cross the membrane. But water can cross the membrane because water is small. And there are little channel proteins, little channel proteins in all cell membranes that allow water to move through. And so let's say water is going to be moving through. I'll get a different color for water. Let's do water in blue. So if there's 10% starch in this cell, doesn't that mean there's 90% water? At least that's what you can think of. I'll do that in blue. And if there's 50% starch in this beaker solution, then doesn't that mean there's 50% water? So wherever the number is higher, that's the higher concentration of water. 90% water is higher than 50%. So water will move from its area of higher concentration to its area of lower. So if we're only considering the movement of water, water will move from the inside of the cell outward. So osmosis will go from the inside of the cell outward, and the cell will shrivel up. It will lose weight, it will shrink. All right, let's set up another one. Let me erase all that, see if I can do it without. Yeah, okay, let's just get rid of all of it. Okay, another beaker question. Practice. All right, let me get my pen colors. Okay, let me make my beaker bigger here. All right, here's another beaker. Here's a cell. Here's the beaker solution. All right, what if there is 1% starch outside of the cell in the beaker solution, and then there is 10% starch inside the cell? But the starch can't move across the membrane, only the water can. Which way will the water move? So what I like to think of is if this is 1% starch, then it's 99% water. This is a good way to start thinking about these. I do that in blue, that's 99% water. If this is 10% starch, this is 90% water. And water will diffuse from its area of higher concentration towards its area of lower. So it will go from the outside of the cell in. So water, if water is the only thing that can move, starch can't, then the water will flow in. And in this case, the cell will fill with water, and it'll either get really full, or if it doesn't have a cell wall, it might even burst. Right, so those are your situations that you could have. Whenever there's a different concentration of solute on one side of the membrane, inside the cell, for example, compared to outside, then you have to look at that solute concentration, but then sort of look at the other part of that, which is usually not given, but it's the water component. And then they'll ask you which way will the water move. And water moves from its area of higher to its area of lower concentration. So the answer is going to be either the water will move from the outside of the cell in, like in this example, or in the previous example, water will move from the inside out. If the water moves from the outside in, the cell will fill with water and maybe burst. If the water leaves the cell and goes out into the beaker solution, then the cell will shrivel up somehow, in some way. It will lose weight, for example. All right, sometimes that kills the cell, sometimes it doesn't. It just depends on the type of cell. But the direction of water movement is something you're going to have to predict and understand why, because it's just following the rules of diffusion and osmosis. All right. So we have terms for the solutions. The terms that we use are comparative terms, and they are hypertonic and hypotonic. These words refer to the solute concentration, not the solvent, not the water. And like I said, typically you're given the solute information, but then you're asked to think about how the water will move. So a hypertonic solution has more solute compared to a hypotonic solution, which has less solute. There always have to be both terms. Even if the problem doesn't say both words, you're always going to have a solution inside the cell and a solution outside the cell. So if they say that the solution inside the cell is hypertonic, that has to mean, therefore, that the solution outside the cell is, in comparison, hypotonic. Because these are comparative terms. It's like saying, you have two individuals, one is taller than the other. You can't just say John is taller. It means John is taller than Cindy. Cindy is shorter than John. So it's a relative term. So if they say you have John and Cindy and John is taller, you automatically know that Cindy is shorter. So these, in the same way, these terms, hypertonic and hypotonic, have to go together. One of the solutions, either the one inside or outside, is hypertonic. The other solution is hypotonic. And so the only other option is if both solutions have equal concentrations, then it's called isotonic. But if you have isotonic solutions, then there's not going to be any net diffusion across that membrane. So that's typically the end point of the process, not necessarily the beginning point, especially for the questions that I'm going to be giving you. And the name of the proteins, the channels that allow water to move through, just for the sake of that detail, it's called aquaporins, which is actually kind of useful. Aqua, it's kind of a water, it has that root of water, and then porins, like a pore, a channel through the membrane. So aquaporins, you've got that term. And those cells have those, and they facilitate diffusion of water, which is osmosis. So the aquaporins are important. All right, so if you have a beaker, and if they tell you that the cell solution is hypertonic and the beaker solution is hypotonic, what they're telling you is where the solute is higher and where it is lower. Now, solute will always want to move from higher to lower concentration, but solvent, remember, goes the opposite way. So water would move into that cell in that example. And there will be practice ones on your post-quiz that you can practice. And read the feedback so that you start to get the hang of it. I'll put in the feedback what the thought process would be as you're solving these problems, and you can practice those as much as you like. Okay, so when a cell is placed into a hypotonic solution, meaning the outside solution is hypotonic compared to what's inside the cell, the water will flow in, and the cell will, like I said before, swell and maybe burst. If the cell has a cell wall, like prokaryotes, fungus, plants, and protists, then the cell will not burst. It will just become very full of water. It will get to a point where no more water can actually get in. But if the membrane can't hold and there's no cell wall, like in animal cells, the cell will burst if the water keeps coming in. So what happens if you put red blood cells in this picture? This is kind of a very standard picture that is used to compare what would happen to animal cells, and they use red blood cells as just an example, but any animal cell placed into a hypertonic beaker solution, an isotonic beaker solution, and a hypotonic beaker solution. And I say beaker solution so you can remember that that's the extracellular fluid. So these describe the extracellular fluid, the beaker solution, in my examples. Then if you put a red blood cell into a hypertonic solution, then the water will leave the cell and the cells will shrink, as in one of the examples I showed you before. If you put a cell into a hypotonic solution, the water will flow more inward and the cells will burst. they can swell, and then if it's very hypotonic, they can burst. So red blood cells, most animal cells like to be in an isotonic solution where water moves in and out at an equal rate, and there is no effect on the cells. However, plant cells are a little different. If you put a plant cell into a hypertonic solution, it too will shrink. It will shrivel up. The cell wall will stay in place, but the membrane, remember, is on the inside of the cell wall, and it will shrivel up. It's pretty cool. You can watch it under the microscope. You can put salt water on a plant leaf and watch it under the microscope shrivel up. In an isotonic solution, the plant is okay, but the cell is not super full of water, like the central vacuole does not fill up with water particularly, and the cell is kind of flaccid, or the cells don't hold any pressure, and so the plant will kind of hang over to the side. And then if you put a plant cell into a hypotonic solution, the central vacuoles will fill up with water, and the cell will be full of pressure against the inside of that cell wall, and we call it a turgid cell. And actually, plant cells are very happy when they're turgid. That means very rigid. So a plant cell is the most happy when it's turgid. An animal cell is happier in an isotonic solution. So those are kind of standard ways of thinking about the differences between cells that have cell wall and cells that don't. So the plant cell could represent any cell with a cell wall. That would be the outcomes for how water will move and what the outcomes will be for the cell. All right, but I will put some practice problems on the post quiz for you to practice specifically. Probably quite a few of them, because that's something you really want to work on. Okay, so I'm going to end this segment, and then if you're ready to go on to the next segment, feel free to do that or take a break.